So in last lecture we discussed about replication rule of 3, where it is divided into 3 parts: APU, central dogma, and assembly, maturation and release. So today we'll discuss about this central dogma, which is nothing but DNA to RNA and RNA to protein synthesis, DNA replication, RNA, and in reverse direction, reverse transcriptase. So this portion will discuss in this lecture. So this is the receptor for the viruses, and this is the virus having some glycoprotein hairs on it, and it bind with the receptors. After this virus is internal receptor mediated endocytosis. Then this virus is inside the vesicle. After this virus genetic material comes out. So these 3 steps, that was the A step, P step and uncoating. So these were the APU step. Once it is released inside the cytoplasm, this is the genetic material. It undergoes, if it is DNA, then will become RNA, then protein, and replication of DNA, RNA multiply itself or it may reverse transcribe. So these are the steps of the central dogma. So DNA and RNA, 2 types of viruses as we know, according to which classification? According to Baltimore classification. According to the Baltimore classification. So these DNA, there are 3 types. We can say there are 3 groups, and these 3 groups are nothing but group number 1, 2 and 7. And this RNA, as per the Baltimore classification, there are 4 groups. And these 4 groups are 3, 4, 5, 6. 3, 4, 5, 6. In which double-stranded DNA, single-stranded DNA, here double-stranded RNA, plus sense, negative sense RNA. So these are the DNA viruses and these are the RNA viruses group. So the usual or general rule for these viruses is DNA viruses in general, they replicate inside nucleus. DNA viruses replicate inside the nucleus, and these RNA viruses, they replicate inside the cytoplasm. This is the general rule. But there are few exceptions that DNA virus, this DNA virus may replicate in cytoplasm. This is the exception to the rule. Exception to the rule number 1, which is the DNA virus which replicate in the cytoplasm is the number 1 pox virus and number 2 is African swine fever virus. So in general, DNA virus replicate inside the nucleus. Exception is cytoplasm, pox and African. Similarly, RNA virus, they replicate in cytoplasm, but there are two exceptions. Exception is RNA virus replicate in the nucleus. So which RNA viruses replicate in nucleus? Number 1 is orthomyxovirus, that is influenza virus. Number 2 is retrovirus. So this is the general rule and these two are the exceptions. But there is one more exception where the virus replicate at both the places, nucleus and cytoplasm, at both the places. And the example is the paramyxoviridae. And one more example is the iridoviruses or iridoviridae family. So this is the central dogma. There are two types of viruses. Proteins are not viruses. We have DNA or RNA viruses. These DNA or RNA viruses, they are classified as per the Baltimore classification. 3 groups of DNA viruses, 4 groups of RNA viruses. DNA viruses replicate inside nucleus, RNA virus inside cytoplasm. This is the general rule. But these two are the exception, that DNA virus replicate in cytoplasm, pox and African swine fever. RNA replicate in nucleus, orthomyxo, influenza viruses and retrovirus. And there are two virus families, they replicate inside both, some part in nucleus, some part in cytoplasm. One is paramyxo, one is iridoviruses. So from DNA to RNA to protein. This is known as replication of DNA. This is known as DNA to DNA is replication of the DNA. So we have some mechanisms through which DNA make multiple copies. What are the replication mechanisms? We have canine parvoviruses. Then we have circoviruses. Parvoviruses and circoviruses. They have different mechanism of the replication. So parvovirus RCR. This is the mechanism which is known as rolling circle replication. Similarly, circoviruses, their mechanism is RHR, that is rolling hairpin replication. So parvovirus RCR and circovirus is rolling hairpin replication mechanism. Then we have papilloma viruses. Number 4 is the adenoma viruses. Their mechanism where the DNA replicate is bidirectional replication. Bidirectional replication. In case of adenoma, the mechanism is known as strand displacement mechanism. Strand displacement mechanism of replication. So from the central dogma, these three part, that is replication, then transcription, then translation. So replication mechanisms are mainly parvo is rolling circle, circovirus is rolling hairpin, papilloma bidirectional replication, adenoma strand displacement mechanism. So these are the replication mechanisms. So before going to this transcription step or translation, we'll discuss one of the virus replication full cycle. Suppose this is the virus and this is the receptor. It comes inside and then uncoating of the viral genome. This is the double-stranded DNA. So this step is attachment, this is penetration, this is APU uncoating. So these are the three steps. Then this is the double-stranded DNA. This is the cytoplasm. This is the cytoplasm and this is the nucleus. So after this uncoating step, this genomic double-stranded DNA, it enters the nucleus. This double-stranded DNA is the parental DNA. This is the parental DNA. So once it enters the nucleus, the first step is the number one is formation of messenger RNA. This messenger RNA is 5 prime, 3 prime, cap, tail, and this is the messenger RNA. And this messenger RNA is early messenger RNA. This is the early one. It comes out and it combines with ribosomes and give rise to early proteins. This is the step 2. And these early proteins are nothing but polymerase enzymes. These are polymerase enzymes. After this, step number 3 is this polymerase enzyme, it acts on this DNA. This DNA make multiple copies. It make multiple copies. And this RNA polymerase, which is nothing but product early protein polymerase number 3, it causes the double-stranded parental DNA to make or synthesize multiple DNAs. So this is the polymerase. The source of polymerase could be number 1 is host cell itself, number 2 is viral early proteins, viral early proteins. So DNA make multiple copies. Then this polymerase, it enter the nucleus, cause this parental DNA to make progeny DNA. This is progeny DNA. Now this progeny DNA, then number 4, it will make number 4 messenger RNA. It will make, but these are late mRNA. These are late messenger RNA. They comes out in the cytoplasm and they make some proteins. And these are known as late proteins. These are known as late proteins. Late proteins are nothing but SP. These are structural proteins. So this progeny DNA will make mRNA with the transcription, and this comes out in cytoplasm, make late protein, structural protein. Now the remaining progeny DNA, these and this structural proteins, again it will come inside the nucleus, and this one both combined, that is known as assembly. That is known as assembly of the, this is DNA and this is the structural protein. Then maturation step, and then steps is the release. Number after maturation there is release. Release may be caused by budding or lysis. So this is one of the replicative cycle of double-stranded DNA virus. After this APU steps, this enter the nucleus. Then the first step is to make the messenger RNA, which is early messenger RNA. It comes out in the cytoplasm, make early protein, which is nothing but polymerase. In case of double-stranded DNA, if it is RNA virus, then here the enzyme formed will be RDRP. Suppose it is negative or positive sense RNA or double-stranded RNA. And if if it is an exception, for example, it is negative sense RNA and it is influenza virus, then that RNA will enter the nucleus and it will form the messenger RNA, some proteins, then proteins will be RDRP, that is viral RNA dependent RNA polymerase, and that will form further RNAs and some structural protein assembly and will release. So the difference between these RNA and DNA viruses is all these either they carry their own RDRP or they synthesize this. For example, negative and double-stranded RNA, they carry their own enzyme, but positive sense RNA, they synthesize their RDRP here. So as a result, all this RNA, they make this enzyme which help in the replication and help in formation of the structural proteins and multiple copies, and assembly, maturation, release will take place. So these are the steps that the replication from parental DNA to progeny DNA. will not take place without the help of polymerase enzyme, which could come from the early protein or from the host cell. Then this progeny DNA will make late mRNA, late protein that are nothing but structural proteins. They assemble with and make virus particle. So these are the steps involved in DNA virus replication. This first step was known as transcription. This was known as transcription. And for transcription to occur, for example, this is 3' to 5' any template. And here we need to synthesize the RNA in 5' to 3' direction. But for this RNA for synthesis, what it needs primer. It needs primer. So primer is the requirement for initiation of transcription, synthesis of new RNA. Synthesis of new RNA, that is messenger RNA in 5 to 3 direction. So different viruses, they use different primer. For example, adenovirus, they use one primer which is here 5' linked, primer, and that is known as 55K protein. Number 2, picornaviruses. Number 2 is picoRNA viruses. They require 5' linked VPG protein. They require this primer for the initiation of transcription. Number 3, orthomyxo, that is influenza viruses. Then arenaviruses and bunia viruses. They need 5' cap from host cell. They need 5' cap from the host cell messenger RNA. And the process they use this cap is known as, these 3 viruses they use this process, and it is known as cap snatching phenomenon. So synthesis of mRNA require primer, and primers are of different types. Like adenovirus use 55K protein as a primer. Orthomyxo, arena and bunia, they use 5' cap from the host cell membrane. And this mechanism of these 3 viruses is known as cap snatching phenomena. They snatch the cap from the host cell messenger RNA 5' end. And similarly, there is one more virus which require the primer, is the retrovirus. Retrovirus uses tRNA of the host cell as a primer. tRNA from host cell. This tRNA from host cell is used as a primer for the initiation of the transcription. So we discussed about transcription. Then what are these early protein synthesis? This process is known as translation. This is known as translation. So translation start from the messenger RNA and this ribosomal interaction. If we look at this diagram of this messenger RNA, five prime cap, this is 5 prime cap, this is untranslated region UTR both side, and this is known as open reading frame. This is known as open reading frame. This open reading frame gives rise to polypeptide chain. Polypeptide chain. And here at the starting we have start codon AUG, and at the end UAA, UAG or UGA. So this is how the messenger RNA is structure is the present. 5 prime cap, both side untranslated region, in between we have ORF, polypeptide chain synthesis from this open reading frame. Some viruses have one open reading frame. Example is the FMD virus. So FMD virus synthesize one polypeptide chain from one ORF. That will further undergo cleavage to form protein like VP1, VP2, VP3, VP4, like this. So one ORF, it form one protein. Some viruses they have 2 ORF, some viruses have 3 open reading frame. 2 ORF they are canine parvovirus, and 3 are calicivirus. So it depends how many ORF. More than one ORF, they will give rise to more than one polypeptide chain. 3 ORF, they may make 3, 4, 5 polypeptide chain which further undergo cleavage, calicivirus. So these are the ORF, open reading frame, single or multiple open reading frame. In case of picornaviruses, in case of picoRNA viruses like FMD or polioviruses, number 1, they lack 5 prime cap. This 5 prime cap is absent, but at this place they have VPG protein. So this cap is absent. At the place of the cap, they have VPG protein. This is in the picornaviruses. Number 3, a long distance between 5 prime and an AUG. Between 5 prime and the first starting codon, there is a long distance. And this, suppose this is the 5 prime and here is the starting codon. In between this portion of the messenger RNA, it is having multiple folding. It is having multiple secondary or tertiary folding due to intramolecular base pairing, and these folding are known as IRES. These structures in between these are known as IRES, internal ribosome entry sites. So these are known as internal ribosome entry sites. Their role is the internal ribosome entry site initiation of the protein synthesis. So this is the unique property in case of picoRNA viruses. They lack this cap, but they have VPG protein, which will be further cleaved by the host cell enzymes. It is removed, but in between these the distance is long, and these base pairs, due to intramolecular bonding, they form one structure known as IRES, internal ribosome entry sites. Their role is protein synthesis. So 5' cap and 3' tail. In between we have OR. So from this mRNA, it may be early messenger RNA, and it may be late mRNA. From early, the protein synthesized are known as early proteins, and here from late, these are late proteins. And early proteins, they are mostly functional proteins, they are mostly enzymes. And enzymes, for example, polymerase enzymes, for example, RNA dependent RNA polymerase. Late proteins are mostly structural proteins. They are structural proteins. They are structural protein means they are capsid proteins. They are capsid proteins. For example, this is the influenza virus, which is having 8 segments, and outside it is having structural proteins, HA structural protein, then M2 ion channel, and below the membrane it is having membrane protein, M1 protein. So these are the late proteins. And inside this core it is having RTRP. Because influenza is a negative sense, these all are negative sense RNA. Means their polarity is 3 to 5 direction. So this is the early protein, and structural protein, capsid protein, they are the late protein. Again, with if we summarize all these steps: attachment, adsorption, penetration, uncoating. After uncoating, we are getting genomic DNA or RNA which enter the nucleus. Then the first step is messenger RNA formation, which is early one. Then it form early protein. This is early messenger RNA, early protein. They enter. These are the nothing but the polymerase enzyme. They enter. They cause this parental DNA to become progeny DNA. These are the progeny DNA. So step 2, protein synthesis. Step 3, it enters. Step 4, that is DNA synthesis, replication from the progeny DNA. Number 5 is the late mRNA. Late mRNA which comes out and it form late proteins, late protein number 6, which are nothing but the structural proteins. They enter the nucleus and they bind with the progeny DNA, and this is the non-infectious assembled virus. So this is known as A. Then it will become mature, become infectious, then it will come out of the cell. That is the release. So we'll discuss AMR steps in the next lecture in detail. But here in this central dogma portion, we discuss the replication mechanisms, then transcription, which is early transcription give rise to early messenger RNA, then late transcription give to late messenger RNA, then translation, whether it is early or it is late, both takes place in the cytoplasm. So early protein synthesis, late protein synthesis, both product they will come inside the nucleus. One will function for the assembly, one will help to for the genetic material synthesis, progeny copies. So in this way, this central dogma, all the 3 steps, replication, transcription, translation occur. In case of retroviruses, they have enzyme reverse transcriptase from RNA to DNA. So they have one enzymes with them and they replicate inside. The different steps for them is they integrate with the host chromosome, host DNA. And in that also formation of early protein, some late protein, then assembly, maturation and release.